Can you see the slides? Can you all see the slides? Gametogenesis?
Not yet, not yet. I
Oh,
think
not yet.
it's coming. It's loading, it's loading.
Why does it tell me sharing is paused until you return something blah blah? Now okay?
No, still not there.
Okay, that's good job.
Okay, screen is sharing.
Okay, now already there?
Yeah. Try
Okay.
opening the slides, doctor.
I already opened, it's on a slideshow.
No, we are looking at what do you call it? Teams app, not the slideshow.
Oh, okay, just a moment. Let me stop. I don't know, I have trouble in this. Just today, lah.
Okay, it's okay.
Let me try. Now is it sharing? No?
Hold on. Usually it will load first. It's not here yet.
Nanti, wait, I'll try again.
Okay.
I'm trying, but it doesn't get loaded. I don't know why. I'm trying to click on the PowerPoint, but it doesn't get loaded. Let me close the PowerPoint.
Yeah, close the PowerPoint and on back.
Maybe I just should leave Teams first and I rejoin again, can I?
You
no because I think something is getting stuck there. Let me rejoin Teams.
Okay, can. Doktor dah keluar ni. Okay, doctor.
Okay, thank you. So thanks for waiting. So today my topic would be on gametogenesis. So do everybody know about mitosis and meiosis and all? Are all you familiar? So you're familiar with it, huh? So
Yes,
okay. So I will just today I plan to complete two topics, both gametogenesis and ovulation to implantation, because I think both are too much interrelated. And when I look back into the timetable, I think next Monday you will have bilaminar and trilaminar. Those are very heavy topics to teach online. So I split it into bilaminar for 2 hours and trilaminar for about 2 hours, so I can give on Friday and Monday. Is it okay? The Friday slot I'm taking for bilaminar. Is it okay? Yes?
Yes, doctor.
Okay, yeah.
Okay, doctor.
Because gametogenesis
Yes,
is known to you all, so I can just breeze through the topic a bit. So the learning outcomes of this session is you will be able to define what gametogenesis is. Gametes are sex cells. This is forming the sperms and the ovum. Genesis is its bond. So how sex cells or gametes are made is called as gametogenesis. We will explain the process of mitosis and meiosis, which help in the process of gametogenesis. Then I always tell you, the process we learn or the concept we learn because we want to relate it to subjects related to medicine. So we will briefly explain the changes during oogenesis and spermatogenesis as well as spermiogenesis. Going to ovulation and implantation, I will be explaining more. So here I will just briefly explain, give an intro to the topic. When I go more to ovulation and implantation, I will explain that further. And you will also have some clinical condition associated with gametogenesis stage. We will see what it is. So gametogenesis, yes, it's a biological process by which diploid or the double number of chromosomes or haploid or the half number of chromosomes. So diploid is 2n, which we call it as double number of chromosomes. Haploid is only n, which is only single half number of chromosomes. They undergo division. Not only they undergo division, but they undergo differentiation. Division means they just divide into different cells. Differentiation means they grow, they become like their adult precursor cells. So division is just like how if a baby is born out of or a small insect, somebody, I mean, if it comes out of an egg or something like that, it is just as a result of division. But once it matures, it changes, it adapts its function similar to an adult life. That is what we call as differentiation. So this differentiation means different division and changing the function to an adult structure is called as differentiation. That results in the formation of mature haploid gametes. So when we start with either 2n number or 46 chromosomes or 23 chromosomes. Then they will undergo division and differentiation to form mature cells, but the final outcome, the cells, is only haploid. They only have half number of chromosomes. Why does the gametes have half number of chromosomes? Why not full?
Because the gamete has to go fertilization.
Yeah, so the male and female gametes fertilize. So finally the full number is restored. So fertilization occurs, mingling of the genetic material occurs. So 23 chromosomes from the father and 23 chromosomes from the mother will unite together to form a 46 chromosome zygote. So if we have double the amount, it cannot happen. So finally, eventually we need only 46 chromosomes. So that's the reason we have haploid number of chromosomes in a gamete. Okay, so how do these gamete cells come? So if you would see this image, the first thing is, where do these gamete cells come from? So in the image on the top, you can see a leech-like structure. That leech-like structure is the embryo. The embryo's abdomen is connected to a yolk sac. So if you would see in a chicken egg, we have the white of an egg and we have a yellow of an egg. Which is the chicken there? Which is the chick there? Have you ever thought of that?
Yellow. The yellow one, right?
The yellow is the food for the chicken. The next time you break open an egg, look for a small red dot or a small white dot near the yellow, very small. That is the chicken. The yellow is the food consumed by the chicken for its nourishment to grow. So the yellow is not the chicken. The yellow is the food for the chicken, and the chicken is a small white dot or a small red dot which you can see when you open the egg. Next time you can see it. Okay, so
Yeah.
so the embryo is just a white dot or a very small dot. So when the fetus is in the mother's womb, it is similar to that of a chicken. You can see only a leech-like structure is there, and it is attached to a big pouch, or what we call it as a yolk sac, because it takes nourishment from the yolk sac. It takes 3 months for the placenta to develop fully, so automatically we would want to take the nourishment from the yolk sac initially, then we rely on the placenta for growth. Okay, so in the yolk sac, in the wall of the yolk sac, the first primordial germ cells are found. So this primordial means they are the precursors. So here in this figure, it is represented by the red color cells. We call them as primordial germ cells. So this primordial germ cells will develop into either a male gamete or a female gamete. The male gamete is the sperm, or the female gamete is the oocyte, which is depending upon the chromosomal sex of the baby. So when we dissect an embryo in the second week of gestation, we can see these primordial germ cells are formed in the wall of the yolk sac. From the wall of the yolk sac, you can see they migrate, they move. In B, they have actually seen the internal aspect of the baby, where I have just circled in a red circle. So these red color cells, they will migrate and come and lie on the red color ridge. We call it as a genital ridge. So the testis and the ovary are formed in the abdomen of the fetus, and once the fetus develops, they will descend. Okay, they will descend. So this is the first step of formation of the primordial germ cells. At this stage, we do not know the gender of the fetus, because we just have only primordial germ cells, and they will just migrate into the genital ridge. So when they migrate, they will undergo mitosis. Mitosis means one cell becomes two, two cells becomes four, and four becomes eight, and so on. So they divide rapidly, they increase in number, and they will move and reside in the genital ridge. Am I clear?
Yes, doctor.
Yes,
Okay.
doctor.
So then they will. So then we want to know, what is mitosis? Mitosis is a process in which one cell divides and gives rise to two genetically identical daughter cells. So we have different stages of mitosis. First, the cell will be going into an interphase. Interphase is where the cell is prepared for division. So that stage is where there is a DNA replication. The chromosomes, which will have only two arms, will start forming another two chromatids. The number of chromosome is the same, only the chromatids are doubled. So that is the phase of DNA replication, and then they will go into prophase, prometaphase, metaphase, anaphase, and telophase. What is the difference between mitosis and meiosis?
Mitosis occurs. Okay, continue.
Yes, yes, yes.
Mitosis produces diploid cells, while meiosis haploid cells.
Very good. Any other differences?
Mitosis occurs in somatic cells,
Yes.
and meiosis occurs in reproductive.
Germ cells. Very good.
Yeah, germ cells.
Yeah. Any other?
Mitosis takes place as only one division stage, like there is six, then that is it. But then in meiosis, it's two stages. There is division one and division two.
Yeah, very good. So these are the basic differences between mitosis and meiosis. Okay, I do have a video on mitosis and meiosis. Maybe I will share that for you now, if I can share, so that you will know the difference. I mean, it is clearer. Wait. Can you see this?
Chromosome, chromatid.
Yes, doctor can.
Yes,
Is
doctor.
the voice heard?
Yeah.
Okay.
Yeah.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid. Written here is 2N. That means it has two sets of chromosomes. In humans, that's including one set of 23 chromosomes from mom and one set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. 46 chromosomes, is the newly duplicated portion is still attached at the centromere region, but there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase and mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible if we say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells. Gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters. A reminder to stay curious.
அதெல்லாம் மஞ்சள் ஐடி வேணுமா?
Sorry, someone mute their mic. Okay, so is it clear so far? Okay, so I felt that when we read mitosis and meiosis separately, it is going to be very confusing because it will actually we will go step by step. So when we go side by side, I think it is easier to understand the difference. Is it clear? Can you hear me?
Yes, doctor.
Okay, so now I will come to the what are the differences between mitosis and meiosis. As your classmates earlier said, mitosis occurs in somatic cells or body cells. Meiosis specifically occurs in gametogenic cells or sex cells, which forms the sperm and the ovum. Mitosis has only one stage and meiosis has two stages. The second stage of meiosis resembles mitosis. So the first stage of meiosis or meiosis 1 is quite special. Why is it special? Number one, because the number of gametes is reduced to half. Chromosomes. So the number is like 46, it is reduced to half, which is 23 during meiosis 1. It is not during meiosis 2. That you have to remember. Then the other thing which is important occurs is crossing over or recombination of the DNA material. So the sister chromatids or the homologous chromosomes. The meaning of homologous chromosome is the chromosomes which are specifically for the hair color, the chromosomes which are for the eye color, the chromosomes which are for the body height, for the body weight. So those kind of things they will recombine. So when they recombine, we get a combination of either the father and mother, or we get one dominant. If the father's eye color is dominant trait, we will go in for the dominant trait of the father, or if the mother's trait is dominant, we go in for the dominant trait of the mother. So this recombination helps us in exchange of genetical material. So at the end of mitosis, the daughter cells are identical structurally and genetically to the parent cell, whereas meiosis, the daughter cells are not identical to the parent cell. At the end of mitosis, we get 2 cells. At the end of meiosis, we get 4 cells. Is it clear now?
Yes, doctor.
Yes, sir. So just go to the YouTube. I think it is Angel something la. You find the video there. So this is what we are trying to tell in. This is what I was trying to tell, but I think the video is explanatory better. So the mitosis, we have prophase where there is condensation of DNA. Then the prometaphase is nothing but there is breakdown of nuclear material and there is microtubules. Microtubules can hold the chromosome in place, and actually they are mobile. So the microtubules are kind of mobile intracellular components. So they will make the arrangement of the chromosomes aligned in the middle of the cell. So this alignment in mitosis is all in a straight line. So later when anaphase occurs, these microtubules will pull each half of the chromatids to either side. So in mitosis, only the chromatids will move, the chromosome number remains the same. Okay, so here next comes telophase, where the spindle fibers are broken up, and during telophase the chromosomes are also uncoiled and nuclear membranes are formed around them. So this is mitosis. So here there is no reduction in the number of chromosomes. When we go to after mitosis is finished, you will have a contractile ring. You can see the ring is formed in the center, so that this the cytokinesis means kind of movement of the whole cell. Cyto is cell, kinesis is movement. So the whole cell will tear apart from each other, move away from each other, split its organelles, and they will form 2 new cells. So this is what happens in mitosis. So in meiosis, it's a cell division that takes place in germ cells to generate male and female. So we have 2 cell divisions, 1 and 2. So one is of course interphase, where there is duplication of the material. So the meiosis 1 is again prophase 1, which happens where there is chromosomal condensation, and during the middle of prophase 1 the homologous chromosome undergoes pairing. This is specific or special for the meiosis. So homologous means the chromosomes that carry the same trait. Example eye color, hair color, the height of the person, the body type of the person, everything, they carry a similar trait. So they are called as homologous chromosome. So there is a recombination, or there is called as crossing over. By pro-metaphase, there is breakdown of nuclear material, and now metaphase is M is middle, as what the video said. So everything is aligned in the middle, but it's not in a straight line. It is arranged in pairs. So when it is arranged in pairs, the individual chromosomes are pulled towards opposite ends. So there is a division of chromosomes here. That's why the cell number becomes half. So here the division or the formation of haploid gametes occurs during meiosis 1 stage itself. Then the last phase is similar to telophase, forming two nuclear membranes and cytokinesis. So now we have two cells with half number of chromosomes. These two cells with half number of chromosomes, they will undergo mitosis now. So meiosis 2 is equal to mitosis. So they will be arranged in the center, they will split the chromatids on either side, and they will eventually, we will have 4 cells with haploid number of chromosomes. Is it clear now? Understandable?
Yes, sir.
Okay. Okay, so now we want to know how we are going to relate this to oogenesis and spermatogenesis. So when we study embryology, embryology is the study of the fetus in the mother's womb. So even before forming a fetus, or even before forming a zygote, the most important function is to form gametes. If we don't have gametes, there is no fertilization. If there is no fertilization, there is no zygote or baby or a fetus formation. So we know that the cells divide by mitosis and meiosis. So now we are going to apply this to oogenesis, or formation of oocyte, and spermatogenesis, or formation of sperms. So first thing is oogenesis. So oogenesis is a process in which the oogonia will mature into mature oocytes. So the mature ovum is called as cyte. Anything with the word cyte is a mature cell. Plus, for example, chondrocyte, osteocyte. When we talk about that, that is a mature cell. Whereas a chondroblast, osteoblast, or oogonia, they are all immature cells. Okay, that's what the term tells you. This oogenesis, it will occur in a female fetus when it is in the mother's womb. For example, consider okay, who do you take in as an example? Okay, take me as an example itself. When does me as a mature woman, when does the oogenesis start? My oogenesis starts when I was in my mother's womb. Is it clear? Whereas when we take a male, that person's spermatogenesis starts only at puberty, around 13 years of age for that person. So the egg or the ova form very, very early in a female fetus. This egg or ova will form very early when the lady is in her mother's womb itself. It's when the lady is in her mother's uterus. Okay, so that's how early it is. So this maturation will start with before birth itself. So we have primordial germ cells, which we saw, which come from the wall of the yolk sac, and they will arrive in the gonad. When they arrive en route, they will undergo a lot of mitosis. So you have hundreds of thousands and many types of oogonia. So that's what we say. The oogonia undergo a number of mitotic divisions. So now we call them as primary oocytes. Okay, so the primary oocyte is actually the ovum cell, and it is surrounded by a flat epithelial cells. So or these cells, which totally we call it as a primordial follicle. So in this picture, if you look, the primary oocyte is just the yellow and the blue. The cells which are forming outside, which forms a row, the squamous cells which form a row around it, is actually the flat epithelial cells. So together we call them as a primordial follicle. Always remember, oocyte needs a lot of support, plus it needs a lot of nourishment from surrounding cells. So it is always surrounded by group of epithelial cells. Okay, so number one, the primordial germ cells, they From the wall of the yolk sac, they go reach the gonad. When they go reach the gonad, by the time the fetus's chromosome is known, either it's a male baby or a female baby. If it is a male baby, all those primordial germ cells will go dormant. They go to sleep. Once the baby is born, once it grows up, once the baby is actually, when it becomes a boy, he becomes a teenager, by 13 or 14 years of age, they open up their eyes and then they start producing sperms, or they change as spermatogenic cells. Whereas if the baby is a fetus, is a female, we know the chromosome is XX, immediately they will start dividing to form primary oocytes surrounded by epithelial cells. So primary oocytes plus the epithelial cells is a primordial follicle. Clear? Clear?
Yes, doctor. Clear.
Okay. Yes, so now, during the next few months, so now the baby is in the mother's womb. It's a female baby. She's developing an ovary, and in that ovary there are quite a few oogonia. When the baby is 5 to 7 months old in the mother's, so meaning the mother is 5 months pregnant, the female baby inside her will have around 7 million oogonia. After the 5th month, the oogonia start dying. Oogonia means nothing but the primordial follicle. So this primordial follicle or oogonia become atretic, means they start dying. And by the 7th month, you only have few surviving. Not few means like almost half of 3.5 million oogonia will be there by the 7th month. So when the baby is inside the mother and the mother is 7 months pregnant, so that baby inside, if it is a female, though all those oogonia will start entering meiosis 1. They will go to the prophase of meiosis 1. So, and they will arrest their cell division in the prophase. So they will stop their cell division in the prophase. Prophase means condensation of the chromosomes. They will stop it there. So what happens at birth? So by 7 months, the baby is having ovaries. The baby will have all primary or primordial follicles with arrested in prophase 1. At birth, all the primary oocytes will go for the diplotin stage. Primary oocytes or primordial follicle means are surround with the surrounding epithelial cells. They will go for a resting phase. So at birth, we can imagine they will have around 600,000 to only 800,000 primary oocytes are usually present. So beginning of puberty, this is approximately reduced to 40,000. So the baby is born. So when the baby is born, if it is a female child, we know the ovary has around 600,000 to 800,000 primary oocytes, or we call primordial follicle with the surrounding cells around it. So now the baby will attain puberty by 12 years of age. So she becomes a teenager. When she goes 12 or 13 years of age, we know that the ovary has only 40 eggs reserve. Throughout the entire life of a person, she will ovulate only 500. So where does this 40,000 go? Any idea?
Menstrual cycle?
Yeah, that's 500. Do we have 40,000 menstrual cycles? So when there is a menstrual cycle happening, the body will recruit at least 15 to 20 eggs, out of which only one is successful to become an oocyte. So every cycle we are actually processing more eggs to release one egg. So that's how the reserve of eggs falls down so rapidly. So most of the time, these eggs which are in the resting or a diplotin stage, they are vulnerable to damage as they age. So that's why there is a lot of chromosomal abnormalities, or the babies has fetal anomalies as maternal age increases. Clear?
Yes, doctor.
Okay.
Doctor, even
Yeah.
if it was 15 to 20 eggs, then it doesn't reach 40,000.
Yeah, but that's the thing. There are a lot of they are vulnerable to damage as they age as well. Two factors are there.
Okay,
One,
doctor,
they are
thank you.
vulnerable to damage. The other one, They are recruited more than what we release.
Thank you, doctor.
Okay, so oogenesis is a complex process which starts when the lady is a baby in her mother's womb. So she will start developing into a primary, when I say primary oocyte, that means only the egg cell. This, when I say a primordial follicle, that means it is surrounded by supporting cells. Okay, so this will undergo prophase 1 and stop in the diplotin stage. Once the lady attains puberty, the girl attains puberty, then she will start developing her oocytes, or they will go into to form mature oocytes or mature egg cells. So there is a drastic reduction in the number of these oocytes from 7 million at 5th month to 600,000 at birth to 40,000 at puberty, and by the time she ends menopause there are no egg cells at all. There are two reasons for it. One, because we recruit more eggs when menstrual cycle occur. Number two, they are more vulnerable to damage. Clear? Yeah.
So the women who have their menstruation cycle earlier, like in their, like when they're 12 years old, so that decreases their age of age that they are capable to give birth.
No, this also depends upon what age the menopause can occur. If they have reserve eggs, so that's why when a lady comes in for infertility, when a couple come in for infertility, we check both the male and female, because we say 33% of the fault lies in the mother, 33% in the father, and 33% is unknown. So we test both of them. One of the tests we look is for the egg reserve. Okay, so it depends, like if she is going to have menopause at quite a number of age, so this also depends upon the genetic factor here. If the mother had a menopause at around 45, more likely the daughter will have menopause at that age. So that also plays an important role. Clear?
Yes.
I want to ask, does the oocytes, like, does the 500 number varies? Like, for example, if the egg is fertilized
Okay.
during puberty, like at the age 12 or 13, does that mean that she will end up having more cells as she grow older?
Fertilized at 12?
Yeah,
Meaning getting a baby?
yeah. So she won't
Actually,
lose more cells.
Actually, yes. Those days, when we were studying, our ONG used to sell. The more the number of babies, then the 9 months your eggs are at reserve.
Oh, okay.
They say. So the more kids they get, you see people are very fertile because their reserve cells are quite more. So we save 9 months. We save 9 eggs actually. Not only nine into around 20, so it's around 180 eggs.
Oh, okay.
Okay, for one pregnancy. So let's come to the maturation of oocytes. So the maturation, so though the oocytes are formed in the mother's womb itself, the maturation or their differentiation begins at puberty, occurs at every month's menstrual cycle. So we are going to select 15 to 20 follicles for one egg. We are going to select 15 to 20 follicles to pass over to 3 stages. Number one, we call it as primary. So we have a primordial follicle. Now we are going to develop into a primary follicle or a preantral follicle. Antrum means a cavity containing fluid. So it is a preantrum. There is no cavity, there is no fluid. So the primary oocyte, what happens is the one which is present in the center, or the ovum egg, which is present inside the primordial follicle totally with the supporting cells, will grow bigger in size. Number one. Number two, you can see the one layer of flat cells will become 3 layers of cells. So these cells are called as supporting cells or granulosa cells. So one layer cell becomes a stratified granulosa cell. So now I call the primordial follicle as a primary follicle. But whatever is present inside this thing is still a primary oocyte. Am I clear?
Yes, doctor.
Okay, so I have a primordial follicle. How do I say it's a primordial follicle? By two factors. Number one, a primary oocyte is sitting inside it. surrounded by a single layer of cells. Once the single layer of cells becomes stratified, but still I have a primary oocyte inside it, I call it as a primary follicle or a preantral follicle. This is the first stage. Imagine a patient, a lady is having a menstrual cycle in January. So she finishes her periods by she has a menstrual starting menses in January 1. By January 5th, she finishes her menses. By January 6th, there is a recruiting of 15 to 20 follicles, and these 15 to 20 will become primary follicles. Then the granulosa cells, what are they? They will rest on a basement membrane, and they will be separated from the ovaries connective tissue. So this granulosa cells along with the oocyte will secrete a acellular layer. In this it is seen as a black arrow, which is we called as zona pellucida. Zona pellucida means lucid, as I said, clear. Zona is zone. It's a clear cell zone layer. It is totally acellular because it does not have a nuclei. The stromal cells or the uterine cells, which is indicated by the blue arrow, will form the theca cells of the folliculi, or the follicles theca or the supporting cells. So this theca cells will have an inner secretory layer or interna and outer fibrous capsule. We call it as theca externa, which is usually not seen in the microscope. We just know they have. So we have a primary follicle. We have a primary oocyte in there. Now it secretes a zona pellucida layer. We have 3 layers of granulosa cells. Outside that we have 2 layers of theca cells. So this is the prime of formation of a primary follicle. Once this stage is formed, we go to the secondary stage or an antral stage. So now can you see a big cavity with fluid there inside it? Can you see that? A big cavity with fluid?
Yes.
So now I just put my laser pointer there, if you can see, okay. So this laser pointer where I've put the red color, that on the surface of the ovary, that is the primordial follicle. Clear? Can you see? So see how many primordial follicles the baby has. Then I am putting my laser pointer now into a medium sized cell in the middle. So that will be the primary follicle. Now I am putting my laser cursor on this big cell with a cavity which is filled with fluid. That is the secondary follicle. Clear?
Yes, doctor.
Okay, so you can see in this one picture a primordial follicle, primary follicle, and a secondary follicle. So second, yeah.
The primordial follicle is one layer of granular cells, right?
Yeah.
Okay, thank you then.
So and they are squamous actually. So the secondary or antral stage is the one which I'm seeing in this picture. It is the longest stage. So it occurs from day 6 of the menstrual cycle. Sampai it will take up to day 12. So it is around 6 to 7 days. As the development continues, you have a lot of fluid pouring. So the granulosa cells and theca cells, they are very secretory in nature. They will secrete substances which nourish the oocyte. They will pour inside to form one big cavity. So that cavity is called as the antrum. This antrum has a lot of fluid. So this, when I see this fluid collection and the oocyte, so here now I'm placing the laser pointer on the oocyte. You can see the oocyte is pushed to one side, surrounded by cells, and a lot of fluid is collected around it. So when I see this stage, I know it is a secondary follicle, but still the ovum inside is a primary oocyte only. It is not dividing yet. It is still a primary oocyte. It is still arrested in the prophase 1. So unless and until it divides, I will not change the name. It is still primary oocyte. So in a primordial follicle, it is primary oocyte. In a primary follicle, also primary oocyte. In a secondary follicle, also it is a primary oocyte. Do you get this point?
Yes, doctor.
Only the surrounding cells will divide and secrete because they want to nourish the egg. Clear?
Clear, clear.
Okay. So now I'm going to the last stage, or the pre-ovulatory. So it is just before ovulation. It occurs if January 1st is the menstrual cycle of the lady. It will occur around January 12 or January 13. So the ultimate maturation of oocytes occur. So pre-ovulatory, we call it as a Graafian follicle. It is the shortest stage, around 37 hours before ovulation. That's why I said day 12 or day 13, because the 14th day ovulation will occur. This is in a normal menstruating female who has 28 days to 30 days cycle. Okay. So at that stage, there is an LH surge, or luteinizing hormone surge in the body of the lady. So out of 15 to 20 follicles which we have recruited, only one reaches the maturation stage. Others will degenerate and become atretic. So now this meiosis 1 will be completed only now. So now I call the egg cell as a secondary oocyte. So when we said meiosis occurs, one occurs, we have two daughter cells. But here, these two daughter cells, one cell will take the maximum amount of cytoplasm. That becomes the secondary oocyte or the egg. The other cell line takes only little amount of cytoplasm, so that becomes the polar body. So out of two cells, we just have one small cell with one small polar body, little amount of cytoplasm, so that dies and goes away. Only the one which takes the maximum amount of cytoplasm becomes a secondary oocyte. So we have a primordial follicle, we have a primary, we have a secondary, and we have a mature Graafian. Primordial follicle is a primary oocyte with single layer of cells. Primary follicle is a primary oocyte with zona pellucida, granulosa cells and theca cells. Secondary follicle is a primary oocyte with granulosa cells, theca cells plus an antrum with a lot of fluid. And a pre-ovulatory Graafian follicle is a secondary oocyte with the polar body with antrum, with granulosa cells, with theca cells. Clear?
Yes, doctor.
Understood? Okay, yeah? So the meiosis 1 occurs pre-ovulation itself. So the maturation of oocytes is like one cell, the secondary oocyte receives most of the cytoplasm, and the other, the first polar body, receives none. It doesn't give much. So then the cell will enter meiosis 2 but arrest in metaphase approximately 3 hours before ovulation. So meiosis 1 is complete. Now they want to divide to another two cells, but approximately 3 hours before ovulation it is stopped. Meiosis 2 is completed only if the oocyte is fertilized. If not, the cell will degenerate 24 hours after ovulation. So, how long does an egg live and how long does a sperm live?
72 hours, right?
Sorry?
For sperm is 72 hours, right?
Yes, usually an egg is 24 hours and a sperm is sperm is 72 hours and egg is 24 hours. Yes.
Okay.
Okay, so we have 146 chromosome, give rise to primary oocyte, undergoes first meiotic division, and then it gives rise to secondary oocyte and the first polar body, and that will stop there at meiosis 2 unless and until it is fertilized. It will undergo another to form a second meiotic division to form a zygote, and then it form another polar body. So here these polar bodies, so we get 4 cells. Eventually we get 4 cells out of one primordial germ cells, but they are mostly polar bodies. You know what's meant by polar body? Polar body is a cell with very minimal amount of cytoplasm, so they are not formed. So one egg forms one oocyte, secondary oocyte, and one polar body. This polar body also will divide into two. So whatever comes out of the polar body it dies and goes away. So eventually only one cell lives even if you have 4 cells. Clear?
Doctor, why
Yeah.
does it have a polar body when during cytokinesis it divides equally actually? Then why there's a difference
In oocyte, no. In oocytes, no. This is one difference here. Sperms, yes. In sperms, equal division. So one cell will give 4 spermatids or 4 sperms. In oocyte, no, because we need only one egg. If at all, if at all they divide equally, they will form 2 eggs. So that's the way where we have what to say, the identical twins. Clear?
Clear, doctor. Thank you.
Okay. So here we have oogonia dividing into 4 cells and giving rise to finally only 1 cell. Okay, so that's what it means lah. So the polar body is actually, it will die and degenerate. So this is the difference. So we started with oogenesis. So please make it clear, oogenesis occurs in a fetus when she is in the mother's womb. If you see a full-fledged female, now you know that her oogenesis would have occurred when she was a baby in her mother's womb. Everything is arrested at the diplotin stage there itself. Only by puberty, they recruit 15 to 20 to undergo primordial follicles, primary follicle, secondary follicle, and a pre-ovulatory Graafian follicle, and they get released. Though the cytokinesis, as what you mentioned, has to have equal distribution of the cell organelles, here there is only one cell which becomes very dominant, the other dies and withers away, which forms the polar body. So please be clear with that. Coming to how we can compare with spermatogenesis. So spermatogenesis begins at puberty. So that's the first difference. So in males, the development of the sperms occurs at puberty only, where spermatogonia form spermatozoa. So the seminiferous tubule, if you have seen the epithelium practical, you saw the seminiferous tubules are arranged in tubes or they are round structures. So I have drawn 2 blocks here. One is black in color, one is having a lumen. So until puberty, all the boys will have tubules, but the tubes is blocked. It does not have a lumen. It is something like a straw with no lumen. Once they undergo puberty, automatically they will acquire a lumen. So where are the primordial germ cells? So the primordial germ cells are sitting on the wall of this lumen of this seminiferous tubule. So those are the red cells or the primordial germ cells which was lining the wall of a seminiferous tubule. So these primordial germ cells will divide by mitosis to give rise to spermatogonial or stem cells. You have 2 types of spermatogonia, type A and type B. By light microscope, you cannot identify what is type A and type B. You just have type A, type B spermatogonia. So this spermatogonia, they are supported by special cells called as Sertoli cells, which nourishes the sperms. So this occurs at puberty. So this formation of type A spermatogonia is what I mentioned. We will have the beginning of spermatogenesis, and the spermatids are embedded in the wall which are nourished by spermatids. So this is the eventual process of spermatogenesis. So what is the difference between spermatogenesis and spermiogenesis?
Spermatogenesis is the formation of sperm.
Okay.
But spermiogenesis is maturation of
So
sperm.
very good. When division occurs, we know it is spermatogenesis. When differentiation occurs, we call it as spermiogenesis. Clear? When I said my first by definition, division is spermatogenesis. Maturation or differentiation is spermiogenesis. So once when you divide, so here when I see this figure, there is a primordial germ cell forms spermatogonia A and spermatogonia B. This spermatogonium then becomes bigger in size and forms a primary spermatocyte. Primary spermatocyte undergoes meiosis 1, reduces the number of chromosomes to form secondary spermatocytes with equal amount of cytoplasm. This then undergoes meiosis 2 to form 4 spermatids with again equal number of chromosomes. So one primordial germ cells give rise to 4 equal spermatids, with equal number of cell organelles and half the number of chromosomes. Whereas in egg, one cell gives rise only to one egg, out of which the three become polar bodies and they don't receive any cytoplasm. That's the difference. Can you understand? Yes?
Yes, doctor.
Yes.
Okay. Okay. So here there are a series of changes resulting in the maturation of spermatids. So I have put three figures here. One is a round cell. The other one, the cell becomes a bit pointed, and the total one, it becomes a sperm. So if you see the first round cell, number 6, or the nucleus, is quite big. Okay, so the spermatogenesis changes or spermiogenesis changes. Number 1, the nucleus condenses. By the time it becomes the sperm, the nucleus becomes small and it becomes more pointed. Okay. Number 2, look at the yellow color structure. The yellow color structure is otherwise called as the acrosome. So in a spermatocyte, the acrosome is just a cell organelle. Now this acrosome will come and arrange itself near the head of the sperm, and it will form like a pointed structure. Why is it pointed and why is it at the head? Is because it will contain the enzymes to penetrate the zona pellucida or the egg. So by right, the sperm will actually enter the oocyte. It will enter into the egg of the female to fertilize. It is not like just they meet together at one point and this one. No, they will actually enter into the cell itself. To enter into the cell, it needs enzyme to eat up the zona pellucida. Those enzymes are present in acrosome, and that's why it is present at the head area. Number 3 is flagellum formation. Why do we need flagella?
For movement, doctor.
Yeah, the sperm is the cell which moves the maximum. It has to move from the testis of the male to the vas deferens, to the urinary tract, come out, enter the female genital tract, travel through the vagina, to the cervix, to the uterus, to the fallopian tube to reach the ovum. Look at the distance it has to travel. So
And
it
what
travels.
did it do?
Yes, it needs a lot of things to travel. So for the traveling, it needs, so that is a life lesson for us. Always have the man has to work to reach to the female. It is not always, it is never the other way around. Okay, ladies, please remember that. Make yourself worthy so high so that the man works to get you all. Okay. So don't go and don't settle for less, ladies. Kamala Harris quota. Okay, we are empowered already. Okay, so to the flagella is formed so that the sperm can move across the male genital tract first and female genital tract to enter the the oocyte, and the most of the cytoplasm is shed. When you look at a mature sperm, there is no cytoplasm. Cytoplasm and cell organelles, they tend to bulk up the cell. So the cytoplasm and fat, everything is gone away, and only the mitochondria are arranged in a row. Why do we need mitochondria?
Produce energy.
Providing energy.
Yeah, they provide them energy to move. Lah. So you need a lot of energy to move. So that's why they have mitochondria. So when a nice rounded spermatid forms a nice long sperm which is fully mature, which is able to navigate through the male genital tract and the female genital tract, and it is able to fertilize the ovum, then we call that process is called as spermiogenesis. Am I clear?
Yes, doctor.
Yes,
Okay. So now let's come to the clinical correlates of this class. So clinical correlates is, number 1 is teratoma. So this baby is got a big tumor in the sacrococcygeal region. It's in the sacral region. It usually occurs in a pluripotent stem cell. We have the three different types of stem cells. We have totipotent, pluripotent, multipotent, and unipotent. Okay. So you can, this is all an extra knowledge for you. If a cell can divide into a fetus and placental material, we call it as totipotent. If a cell can divide into 3 germ cell layers like ectoderm, endoderm and mesoderm, we call it as a pluripotent. If a cell can give rise to one ectoderm and different cells in that, we call it as multipotent. Like for example, ectoderm can give rise to nervous tissue and skin. So that is a multipotent cell. And if a cell can give rise to only one group of cells like skin cells or nerve cells or muscle cells, that we call it as unipotent. This is an extra info for you, so if you want you can go read more on that. So these teratomas come from pluripotent cells, so meaning they give rise to 3 germ cell layer cells. So usually these are from the primordial germ cell. So when they move and they go to the gonads of the fetus, they will automatically divide into 3 different types of cells. That's why they're found in that particular area. So they will have a variety of tissues like bone, hair, muscle, gut epithelia and things like that. When a cell becomes like primordial germ cells, it becomes abnormal and migrates. It divides also abnormally, and that's why it gives rise to all these different kinds of cells rather than gonadic cells. It has to be in its right place to divide and form gametes or gonad cells. So in this case, the prognosis is quite good. If a baby has this acrococcygeal teratoma, it is excisable and the prognosis is very good. So it's nothing to worry about the baby in this case. So the last part of the lecture is chromosomal abnormalities. We talked a lot about chromosomes, how they divide, how they form diploid, haploid, everything. So here I put a lot of features, blah, blah, blah. Don't get scared of it. For me, for you to understand chromosomal abnormalities, I want you to understand only 2 things. There are numerical abnormalities and there are structural abnormalities. Okay, numerical means abnormalities due to changes in number. Structural means abnormalities due to change in structure. Please don't force yourself to read, okay, Down syndrome, growth retardation, epicanthal fold, simian crease, heart anomalies. Don't. The most important thing in Down syndrome you're supposed to know is it a numerical anomaly or is it a structural anomaly? If it is having problem with numbers, I call it numerical. So the baby will have these defects. Okay, something like that. So the first numerical anomaly we see here is Down syndrome. It's very common. Down syndrome is very risk increases with increased maternal age, and I think you know why now, because in the diplotin stage there is a lot of propensity to damage the oocyte, and the oocyte has been with the lady since she was a fetus. So you imagine the amount of radiation, the amount of I mean, even foods have radiation. Actually, banana is a food which has quite high radiation, you know. So starting from food with electromagnetic radiation, the amount of X-ray, the amount of CT we do, the surrounding, the laptops, the gadgets we use. So look at the amount of radiation we have around us. So this can affect the oocyte, and eventually as the mother ages, there is, so usually after 40, usually there is very high risk of Down syndrome. When the lady is around 25, the risk of Down syndrome is around 1 in to 400, something like that. But when it goes to above 40, she becomes, the Down syndrome is 1 is to 50. So the risk becomes very high. Okay. So there is an extra copy of, so don't go tell me, so this one don't go to your parents. They say my lecturer asked me to get married earlier. La. You don't go tell your parents and quote my name. Huh? I'm just teaching medicine. Okay, so there is an extra copy of chromosome 21. This is because there is non-disjunction and it's primarily formed in the oocyte formation. So the risk of Down syndrome occurs as the mother ages. So usually when a lady is pregnant after the age of 40, they usually do 2 types of tests. They do triple test for Down syndrome, because usually there is a type of hormone that is increased in the mother, like alpha fetoprotein, and they do a triple test for that. If there is any abnormality in the triple test, then they will do an amniocentesis to type the chromosome of the baby. If they find it like 21 trisomy, automatically they will tell by even before birth, or they do a chorionic villus sampling. Dr. Sarah will talk to you next week about those investigations. So that stages we can detect the Down syndrome. If there is a 3 copy of chromosome of the 18th chromosome due to non-disjunction, we call it as an Edwards syndrome. So Edwards syndrome is nothing but there is a 3 copy of chromosome 18. This chromosome is very important for the phenotype. So you can see the baby has a big head or dysplastic ears and things like that. And if there is a trisomy 13 or 3 non-disjunction chromosomes, we call it as a trisomy 13 or Patau syndrome. So at your stage, I think Downs is quite important for you all to know. It's trisomy 21 because it is proven to be because of maternal disjunction of the oocyte. Whereas Patau syndrome or Edwards syndrome is not that common. We don't see that much. If there is a non-disjunction that occurs in the sex chromosome, the cells will have 47 chromosomes. So that is called as Klinefelter syndrome. So the genotype of the boy is XXY. So actually it is a boy, but they will have an extra X chromosome. So here they will have a female pattern genitalia or pubic hair pattern. They will have some amount of mammary glands development, and if they might have some amount of ovarian tissue inside, and they will not show it outside, they might have some amount of ovarian tissues. So in Klinefelter syndrome, if we diagnose a Klinefelter syndrome, usually they will be given a choice whether they want to they change their genetic identity or something like that. If they want to remain as males, the ovarian tissue will be removed. They can be given some male hormones to make them feel more like males, but still their chromosomal pattern is X46, XXY. Always it is like that. Okay. Then we have Klinefelters. It depends. Klinefelters, because they have an extra X. It depends on the hormonal level. Because spermatogenesis is basically on. It starts at puberty. We still have the chances of reviving their I mean, prevent them being infertile, but it's pretty difficult because still their chromosome remains the same. Okay. Then we go to triple X syndrome, which is again the 23rd or the sex chromosome, but we have triple X. So these are tall. Usually the female child is having low IQ and they will end up having some limb defects. So 47 XXY is Klinefelter's. That's very common. Triple X is not that common, which is just an example for you. And the next is with this one is Turner syndrome. Turner syndrome also is quite common. So here one X chromosome is missing. So we saw Downs is trisomy 21, Edward is trisomy 18, and Patau is trisomy 13. So extra copy. If it occurs in the sex chromosome, it is Klinefelter's and triple X. Now we come to Turner's. The number is reduced. So it is 45 only. So the sex chromosome supposedly should have another X. There is no X. So the second sex chromosome is absent. So this Turner syndrome, they will have short webbed necks, and you can see the Turner syndrome. Very easily the phenotype is identifiable. So out of structural abnormalities, Turner syndrome is the one which the number is less. Not structural, numerical. Turner's is the one which has the number less. All others have number higher. Then coming to the next one is your structural abnormalities. So this is, the number is correct. The person has 44 plus XY or 44 plus XX, but they will have structural abnormalities because of breaking of chromosome or deletion of the broken piece. Number one is Cry-do-chat syndrome. The baby cries like a cat. Okay, that's why they call it as Cry-do-chat syndrome. Here, there is a deletion of chromosome in number 5 in the short arm chromosome. It usually leads to mental retardation and heart diseases. Number 2 is Angelman syndrome, and 3 is Prader-Willi syndrome. If there is a micro deletion in maternal chromosome, that's why they call it as Angelman code, or it is found out by Angelman actually. If it is a deletion in the maternal chromosome, they call it as Angelman, and if there is a deletion in a male chromosome or the paternal chromosome, they call it as Prader-Willi syndrome. The next one is Fragile X syndrome. Fragile X is either transmitted through a male or female, but this is very commonly found in males because females have another copy of X, whereas males have only one X. So if they get the defective X, it is more clearly evident in them. So that's why males are affected more. Females also will be affected, but since they have two X, It doesn't matter, the other one will be dominant. Here, the male, since they have only one X, if it is deleted, automatically it is more pronounced in males. So this Fragile X syndrome is included in a part of autistic syndrome. So they are kind of learning and cognitive disabilities. They don't not totally mentally retarded, they might have cognitive disabilities or a part of dyslexic syndrome. So that's all for this class. So as for the syndromes which I have talked to you, first, divide them into numerical and structural abnormalities. What is in numerical? Divide them into one which increases, one which decreases. So just put trisomy 13, trisomy 18, trisomy 21. So it would be the Edward Patau and then Down syndrome. Then go through the sex chromosome defects would be the Klinefelter's triple X, and if it is reduction in the number of chromosome, it is Turner's. When you go to structural abnormalities, it will be Cry-Duchat, it will be Angelman, Prader-Willi, or it will be Fragile X. So just understand what is the defect in that. That's all. I'm not going to ask you like, hey, the baby has this, what is the reason? That all you will learn in pediatrics. You still have a lot of time to learn that. Here we are just going to relate how this chromosomal abnormalities can cause abnormalities in the fetus. That's all. Any doubts in this topic? Umar, are you in?
Hi, Doctor. Yes, I'm in.
Yeah, please put the attendance into the chat.
Okay.
Do you want me to continue with ovulation and finish it by 10:30? Or
It should be no problem.
can I? So I will give you a 5 to 10 minutes break maybe. I will start around 9:35. Just put just reminding the class, the attendance has already been put into the chat, and please make sure you sign your attendance now.
Okay, doctor.
Okay, please don't bug Dr. Atika for it. So please sign your attendance now. I'm reminding you once again. So I will just start the class around 10 minutes time. Is it okay for all of you? Okay?
Yes, doctor.
Yes, doctor. Okay.
Okay, yeah. So just take a 10-minute break, because I think the ovulation and implantation is a continuation of this. So it is easy for you to understand that if I continue that way. Okay? So just take a break. We'll see you all then. And don't forget to sign your attendance, please. This is the third time I'm reminding you. Please sign your attendance.
Doctor, should we assign our attendance for our next topic as well?
Just for this. For Friday, she will give you an attendance sheet, and Friday, anyway, I'm going to have a class for you. This is just, I'm taking the extra time for this class because Monday's class I'm splitting into two. It is difficult for me to explain in 2 hours bilamina and trilamina because it will take some time. This has been quite easy for me because you already knew gametogenesis. If it's bilamina, trilamina, it's going to be heavier. So that's why.
But doctor, we have to. So that means we still have to submit 2 attendance for today, right? Because we have to enter the subject, also the topic.
Did she give you 2 forms?
No, actually, she gave us only one yesterday.
Okay.
But it's only for this week and next week. So meaning we have to repeatedly fill the form, but every time we fill the form, we fill in different topics.
Okay, now you can fill it up. So for this one, you just fill it up as gametogenesis, and then you fill in again for ovulation, twice.
Okay, thank you, doctor.
Okay, yeah, okay. But don't worry, if some people fail to fill ovulation, I will just tell them to take the gametogenesis because I finished it in one class. I will inform Dr. Atika on that in case people are missing it out. So please make sure at least you fill in one form. Okay. Can I start now?
Yes, doctor, go ahead.
Okay, so again a reminder, please sign in your attendance as what Omar said, maybe twice with the name of the topic which you attended to. So today you're attending two topics, gametogenesis and ovulation to implantation. I have already informed Dr. Atika that I'm going to cover these two topics because they are quite closely related. So she will take either or attendance for both. So nothing to worry, I will tell her. La. Okay. So now coming to ovulation to implantation, actually we saw most of the ovulation features what we talked about. So just a little more elaboration is needed in this topic. So that's why I decided I want to do it together. So ovulation to implantation, the learning outcome is you're just going to briefly describe the ovarian cycle and the hormonal regulation of ovulation. You will learn this a lot during the reproductive system. So this is just the beginning only. Yeah? So we're just going to briefly describe as well as the process of ovulation and the changes in the endometrium. This again, you will learn it in the reproductive system. So just for the introduction only, I'm going to tell you. And then the precondition of the sperm for fertilization. So always the sperm, as soon as it gets released from its male genital tract, it is not ready for fertilization yet. It has to undergo a lot of preconditioning for fertilization, and explain the three phases and results of fertilization, what happens. And we will have there is a structural changes from the zygote. Zygote is nothing but the fertilized fetus, which is a fertilization of the sperm as well as the egg. So it will transform to a blastocyst. So when I continue, I will continue with the blastocyst. So from blastocyst, how it becomes a full-fledged fetus, until then we will finish it by next week. So when the baby is already formed, it cannot just be dangling inside the uterus, it should be implanted into the uterus. So we want to know the normal process, how it is implanted and which site it should be implanted. An abnormal implantation, we call it as an ectopic pregnancy. So now we are reading the process of ovulation, the fertilization and implantation with clinical condition. We are reading processes, three processes: ovulation, fertilization and implantation. Anything that goes wrong in these processes, we will have clinical conditions. Plus I will touch upon the artificial reproduction techniques which we use in vogue nowadays. So what happens during puberty? At puberty, so we are starting with females. So at puberty, the female begins to have menstrual cycle. So at that point, the hypothalamus will release gonadotropin, releasing a hormone. So gonadotropin means the sex hormones. So it will secrete gonadotropin, releasing hormones, which will go to the pituitary gland. The pituitary gland is situated at the base of the brain. So once the pituitary gland is stimulated, it will secrete two types of hormones. One is luteinizing hormone, the other one is follicle stimulating hormone. So both luteinizing hormone and follicle stimulating hormone are present in both male and female. But in males, this luteinizing and follicle stimulating hormone are usually present at a baseline level throughout. Whereas in females, there is actually a waxing and waning, increasing and decreasing, because the female, the changes are occurring every month. So if first we say we want to recruit 15 to 20 follicles, the follicle stimulating hormone will be very high. Once ovulation wants to occur, the luteinizing hormone, there is a surge. So that's why we say the female has hormonal changes during the menstrual phase, and that's why they become quite moody as well. So males do not have a reason to become moody. Yeah? Females have all the reasons to become moody. So at puberty, so the female will usually have both male and female, they will have secretion of GnRH, but here I'm just concentrating on the female only. So they will secrete GnRH, stimulate the pituitary gland to produce FSH and LH. If these hormones stimulate boys, they will produce testosterone. If they stimulate girls, they will produce estradiol and progesterone. So this is the changes that occur during puberty. So coming to the ovarian cycle, what is the hormonal control of a ovarian cycle? So as I said, January 15, January 1, January 13 to 15 and January 28 to 30. That will be the menstrual cycle of the lady. January 1st, just look at the green color. The follicle stimulation is quite high. So now the ovary will start recruiting 15 to 20 follicles and they will develop. These follicles will secrete estrogen, which is the blue color or the indigo color la. So that is the blue color. So now you see the estrogen is quite high. When automatically it goes to mid phase, that is January 13 to 15, there is an LH surge which is seen in red color. So when there is LH surge, out of 15 to 20 follicles which have been recruited earlier, we form primary, we form primordial, primary, secondary, 15, only one becomes the Graafian follicle. So that Graafian follicle, it ruptures, it comes out of the ovary and it will be waiting for fertilization. That process is called as ovulation. So once ovulation occurs, the remaining granulosa cells which are surrounding which are left over back in the ovary itself will start secreting progesterone. So that's the progesterone. The cyan blue color is the progesterone. So in the early stage, the progesterone is at a baseline level, and in the later stage, the progesterone becomes quite higher. The last pink hormone is HCG. HCG is human chorionic gonadotrophin. It is also present at baseline level, but if the oocyte is fertilized, it will increase because it is secreted by the zygote. If it is not fertilized, the HCG remains at the baseline level. Am I clear? Clear?
Yes, doctor.
Okay,
Clear.
just a moment. Just a moment. I'm just getting a call from the university. I need to take up the call. I will be back. Okay, I'm back. Thanks for waiting. Okay, so we just discussed the ovarian cycle where the green follicle stimulating hormone begins starts increasing at the level of January 1st. But the lady is still in the menstrual phase, mind you. So when it increases by January 5th, it will recruit 15 to 20 follicles that secrete. You can see estrogen starts increasing only by the 5th day. So when it increases, then when all the follicles are ready, there is an LH surge, which is a peak. The red color peak is there, and that releases the Graafian follicle. Once it is released, the remaining cells or the granulosa cells, now they will secrete more of progesterone. Progesterone is the maintaining hormone. Why we call as maintaining or maintenance hormone? Because it causes a lot of water retention. You know, the second half of the menstrual phase of a lady, there is a lot of water retention. There is a lot of tender, sore mammary glands. There is, you feel bloatedness, because basically we are retentioning water, making the body accommodating to implant the baby. The body doesn't know fertilization occurred or not. Only we wait until the end of the January 28, if there is no fertilization occurs, there is no HCG, automatically the progesterone level drops and then menstruation occurs. So this is the hormonal control of a ovarian cycle. So going further, the ovarian cycle, that's what I mentioned. In the days preceding ovulation, the secondary follicle will go up to 25 millimeters. So in the mid-cycle, there is an LH surge, or there is an increase in luteinizing hormone. So what does this LH surge do? It will cause the oocyte to complete meiosis 1. So we already, they are stopped in the metaphase of meiosis 1. They will initiate meiosis 2. But still this meiosis 2 will be arrested in metaphase 2. So the secondary follicle, it will form one Graafian follicle with a secondary oocyte just 3 hours before ovulation. It will stimulate the production of progesterone by the stromal cells. Stromal means the cells or the theca cells or the ovarian cells that surround the follicle. So that is what we call it as stromal cells. And eventually it will cause the rupture of the Graafian follicle and release of the oocyte. We call that process called as ovulation. So LH is a very important hormone for ovulation. It will complete the division, stimulate the production of progesterone, making the body ready for acceptance of a fertilized zygote, and it will cause ovulation. So this is a picture of ovulation that has been taken during endoscopy from a female. So during ovulation, you can see the ovary begins to bulge. The red area is a part of the wall of the ovary where there is bulging. And the apex or the tip of it where the yellow color substance is present, that is called as the stigma. So now that at that area, luteinizing hormone will increase the collagenase activity. Collagenase is an enzyme which digests the collagen. So it will eat away all the collagen surrounding the follicle. At this point, the lady also secretes prostaglandin, so it will produce local muscular contraction to push out the oocyte. Mind you, the oocyte does not have any flagella to move. So it is the purpose, the LH, the muscular contraction, everything have to just milk out the oocyte. They cannot move by themselves. So during ovulation, there is a bulging of the wall of the ovary. There is increased muscular contraction. There is digestion of collagen surrounding the follicle. So this follicle, if you see, the oocyte is in the center. The surrounding yellow cells are the granulosa cells. They radiate, or they are around, or they are found all around the oocyte, and that's why they are called it as corona radiata. So these cells, why does the oocyte have corona radiata? Because it needs constant nourishment. So it is pushed out of the ovary, but still it needs nourishment. So it takes a group of cells surrounding it. They call it as corona radiata. Am I clear?
Yes, doctor.
Okay. So now the lady has pushed out, ovulation has occurred, the lady has pushed out the oocyte along with the corona radiata. What happens to the rest of the cells that were filling up the Graafian follicle? So under the influence of this luteinizing hormones, the remaining granulosa cells within the ovary, they take a yellow pigment color and change. So when they take a yellow pigment color, we call them as lutein cells. The yellow pigment is because of increased amount of cholesterol. All sex hormones are cholesterol based hormones, so they will take up the yellowish color very well. And now it is called as corpus luteum. Corpus means body, B-O-D-Y, body. So when there is a body of yellowish color cells, we call them as corpus luteum. So when we see an ovary of a lady who is just ovulated, she will have a big corpus luteum. So the ovum has come out. It is waiting in the fallopian tube for fertilization. In the ovary, the rest of the cells which were surrounding it, forming the Graafian follicle, will acquire the yellow pigmentation because of cholesterol, and they will appear as yellowish body called as corpus luteum. This corpus luteum secretes little amount of estrogen and a lot of progesterone. So progesterone secretion is the main function of corpus luteum. So these hormones will induce the ovary to the uterus to enter the secretory stage. So these hormones are immediately spilled on into the blood. So because they are endocrine hormones, they are taken to the uterus. So now the uterus will, the glands of the uterus will become bigger. It is waiting for the zygote to come and implant. So if you would remember, we recruited 15 to 20 follicles. Only one become a Graafian follicle. So what happened to the rest 14 to 19? So the other follicles, the 14 to 19, they will degenerate, they will become atretic, and we call them as corpus atreticum. Clear? Clear?
Yes, doctor.
Okay, so at the beginning of an ovarian cycle or a menstrual cycle, we are recruiting 15 to 20. Only one will reach the Graafian follicle stage. And once the ovum oocyte is given out of the Graafian follicle, the rest of the Graafian follicle forms corpus luteum. The rest 14 to 19 which we developed, but they will go atretic. They will die off and they will form bodies we call as corpus atreticum. So usually there is a connection between an ovarian cycle and a uterine cycle. So when you look at the graph on top, the FSH is increased, so you are recruiting the follicles. When there is an LH surge, automatically ovulation occurs. The rest of the things form corpus luteum, and this corpus luteum, after it gives around all the hormone, it becomes whitish in color. We call it as corpus albicans. Then again the cycle starts all over again. See, again the FSH will increase, the blue color will start increasing. So these are the changes that occurs in the ovary. Let's look at the hormonal changes. So when there is an FSH and LH stimulation, there is increase in estrogen in the earlier phase and increase in progesterone in the second half of the menstrual cycle. This is secreted by the ovaries. So this will be going and stimulating the uterus. So when I go down, look at that shows the uterine wall. So the first is menstruation occurs. After menstruation, there is follicular stage where there is developing follicles. So the wall builds up slowly. By the time ovulation occurs, progesterone secretes more. Now the progesterone will tell, prepare the uterus, the zygote may come for implantation. So the uterus increases the number of glands for secretion, increases the blood vessels and keeps it ready for the baby to come and attach to itself. If the fetus is not attaching, there is no use for it. The progesterone falls down and whatever we develop, it is shed and come out as the menstrual fluid. Clear? Clear?
Yes, doctor.
Yes, doctor.
Okay, I think this you already learned before. So now we know that the oocyte has been released. Again, I told you the oocyte does not have a flagellum to move. So the fimbria or the finger-like processes of the uterine tube, they will actually sweep over the surface of the ovary. So the oocyte with the surrounding cells will be taken into the tube. So the oocyte is barely propelled by two things. One, the peristaltic or the movement of the muscular movement of the fallopian tube and the cilia. So this is a place where you have cilia. Another place where you have cilia? Another place or any other epithelium where you have cilia?
Trachea.
The windpipe.
Yeah.
Trachea's
Yeah.
reproductive tract.
Reproductive and
Respiratory tract.
respiratory. Yeah, so in the respiratory tract we move the mucus or what we call as hingus in Malay, but the mucus, we call it mucus in English. So we move that mucus in the respiratory tract, whereas in the reproductive tract the cilia are used to move the zygote or first the oocyte, later the zygote. Okay. So this, in case fertilization occur, the fertilized oocyte reaches the uterine lumen in about 3 to 4 days. So the fertilization usually occurs in the fallopian tube. Okay. So what is corpus albicans or corpus luteum? So what is left over? If the oocyte is not fertilized, the leftover cells or the corpus luteum will develop its maximum development in 9 days. After 9 days, it will give away all the progesterone, then it will become, so once it gives the progesterone, everything. The yellow color is lost. Now it becomes white in color. Alba means white. That's why we call albinism is white. They don't have melanin. We call them as albinism, the condition. So once it gives away all the progesterone, the corpus luteum takes up a whitish color. So now it becomes corpus albicans. If fertilized, the zygote will secrete human chorionic gonadotrophin, or the baby itself will secrete a hormone. This helps in maintaining the corpus luteum. So in that case, if it is fertilized, the corpus luteum keeps on secreting progesterone till the 4th month of pregnancy. So the early stages of pregnancy, the corpus luteum is very, very important. In case the corpus luteum dies or degenerates in a pregnant lady, the pregnancy also will die. She needs progesterone produced by the corpus luteum to maintain the pregnancy. So the early pregnancy maintenance is because of hormones. Only by the 4th month, the placenta will completely take over. Still, they need progesterone. They will be still secreted by the cells of the ovary, but not as high as they need at the first 3 months of pregnancy. So by 4th month, the placenta will replace the nutritional support, the metabolic support to the baby. So automatically the baby grows in size. Until 3 months, the growth of the baby is not much, but there is a lot of differentiation. We form most of the major organs by 3 months of pregnancy. That's why they say the early stages of pregnancy is very important. If you're exposed to radiation, if we take any drugs, which is not good for the fetus, it will lead to congenital anomaly of the baby. Am I clear?
Yes, doctor.
Okay. So now the ovum or oocyte has been released. It is waiting in the fallopian tube. So this occurs, then fertilization occurs. So fertilization is a process by which the male and female gametes fuse. So this occurs in the ampulla. Ampulla is actually the widest part of the uterine tube. It has 4 parts. We have, I don't think so that's important for you now. It's for you, it's just enough if you remember it occurs in the uterine tube. Only 1% of the sperms which enter the female genital tract will enter the cervix, where they survive for many hours. Usually a trip to the cervix to the uterine tube takes about 2 to 7 hours for the sperm to navigate. So when ovulation occurs, this oocyte along with the corona radiata will secrete chemoattractants. These chemoattractants will pull the sperm, or it will attract the sperms towards the fallopian tube. So all the sperms will basically swim towards the particular tube. So when the sperms enter the female genital tract, they have to undergo a process called as capacitation. Same thing like real life lah. It's not necessary you're a male, you just go propose a female. You have to have a job, you have to have a house, a car, hantaran duit lah, inilah. You have to be able to satisfy the female both by her, or whatever needs she has, you have to give her. So you have to make yourself capable of even proposing to a female. Isn't it nowadays like that? Not nowadays, always it's been like that.
Correct.
Yes. So this same thing happens in our body also. So the sperm has to undergo 2 processes to acquire the ability to fertilize. These 2 processes are called as capacitation and acrosomal reaction. So they have formed from a spermatocyte, they form a spermatid and a mature sperm. But these sperms have to have the capacity to go to fertilize. One is it has to swim faster. Number 2, it should be able to develop a good acrosomal cap to penetrate the oocyte. So what are the changes? Number 1, capacitation. Capacitation and acrosomal formation occurs in the female genital tract. It never occurs in the female. So this is a conditioning in the female genital tract of the sperm, especially it occurs in the uterine tube for about 7 hours. The sperm usually has cholesterol embedded coating in the outside layer. This cholesterol is actually a protective layer. Like how I say, adipose tissue is very protective and insulation. So this cholesterol prevents the killing of the sperms by the acidity in the vagina. So once they reach the cervix and the uterus, they don't need the cholesterol, so the cholesterol coating is lost. There is a hyperpolarization of the sperm plasma membrane, so meaning it becomes very fast and there is a hyperactivity. So they start moving at a very fast rate and they respond to the chemoattractants given by the oocyte. So these are the changes that occur in capacitation. What happens in the acrosomal reaction? So here acrosomal reaction occurs only if the sperm binds to the zona pellucida after the sperm meets the egg. So the zona pellucida will induce the acrosomal reaction. So you can see the head of the sperm usually has glycoprotein coat, plasma proteins. So it will remove all, it will digest away all these areas, and acrosin and trypsin like enzymes are present. They are protein enzymes. They will digest protein. You said zona pellucida is an acellular protein layer. So it will digest the zona pellucida and it will enter into the oocyte. Okay. So we have talked about how we have changed from a primordial, primary, secondary and a Graafian follicle in a female. The Graafian has released the oocyte. The rest of the follicles form corpus luteum. The other follicles which we develop form corpus atreticum already. So the oocyte is waiting in the fallopian tube, especially in the ampulla of the fallopian tube. So the sperms which have been released into the female genital tract undergo a process of capacitation, number one, meaning it will become hyperactive, remove the cholesterol, and it will move or swim towards the respective tube. When this one sperm meets the egg, it will undergo acrosomal reaction induced by the zona pellucida, where it removes the head or the glycoprotein and seminal plasma protein, and it will secrete enzymes to digest the zona pellucida itself. So basically, if you have a lot of head weight or ego, no lady will respect you. Remove it, remove your head weight or helmet or your turban and then go to females, they will accept you. Life lesson. Correct?
Correct.
Correct, yes.
Yes.
Oh my God, I'm going to get hold of Dr. Reshma as a feminist. Nola, this is what I learned from this. When I read this, usually I will relate it to whatever happens in real life. So it's like that. So those days, when they want to marry a lady, like those hunting days, they will hunt. They will break big things. They will lift big stones to show their strength. That's how it used to happen. It is not like nowadays now. Those days it was like that. Men have to show their power to go and get married. If you see the tribals, still they have a lot of games to attract females. Whoever is the winner, they will be given the lady or something like that. So that's the principle behind this law, which we read it in embryology. So now the sperms have the capacity to for fertilization. So we talked about fertilization. So what are the three phases of fertilization? Phase one is penetration of corona radiata. So it penetrates the surrounding cells. Phase two is penetration of zona pellucida or the acellular layer. And phase three is the fusion of the oocyte and the sperm cell membranes. So we might think just because meeting of the sperm and egg it's fertilization. No. It is first entering the corona radiata or surrounding cells, entering the zona pellucida or the protein layer, and phase three is the oocyte and the sperm cell membranes. So first one, out of 200 to 300 million sperms that deposit, only 300 to 500 reach the site of fertilization. Of this, only one fertilizes the egg. Okay, so that is the penetration of corona radiata, Sato. Number two is the penetration of zona pellucida. So we said zona pellucida is the glycoprotein shell that surrounds the egg that facilitates sperm binding to induce acrosomal reaction. So now the sperm head, once you remove the glycoprotein, the plasma proteins, there is the acrosomal cap is exposed. When it is exposed, it will release acrosin. It allows the sperm to penetrate the zona. Again, now when one sperm penetrates zona pellucida, all the cortical granules in the cytoplasm of the egg, they will know, okay, one sperm has penetrated. So now the egg becomes hostile to the other sperms. Same thing, like if a lady is married to a man, after that she will not go around pampering around telling, okay, I'm still free. She is married la. So similarly, when the penetration of one sperm occurs, the cytoplasm of the oocyte will recognize it. It will start releasing lysozymes to change the configuration of zona pellucida. So the other sperms will come and try to enter. It will not be entering. So this is a prevention of polyspermy. So this makes the environment hostile for the penetration of the zona by other sperms. Okay, we do have conditions where there is a polyspermy where two sperms enter. They can actually fertilize the egg, or we can end up having multiple in case. But usually the cases of twins is where we have two eggs are released and it's fertilized by two sperms, or a zygote is formed and it divides into two babies. So that is the most common causes rather than polyspermy causing twins. So most common is either the zygote itself divides into two, or two sperms, they fertilize two eggs. So that's the most common causes of twin pregnancies or multiple pregnancies. The last phase is the phase 3 of the oocyte and the sperm cell fusion. So here the head and the tail of the spermatozoa will enter, only the plasma membrane is left outside. So when it enters, now we can only see the nucleus of the oocyte and the nucleus of the sperm. So they become what we call it as pronuclei. So here, okay, so just the three phases again I will repeat. Number 1, penetration of corona radiata. Number 2, the sperm penetrates zona pellucida by releasing acrosin and trypsin like enzymes. Once it penetrates, the egg becomes hostile to the other sperms. Number 3, fusion of oocyte and sperm cell membrane will occur. So just the cell membranes will fuse and the sperm itself is included into the oocyte, leaving its plasma membrane. So as soon as the sperm enters the oocyte, will respond in 3 ways. Number 1 is the cortical and zona reaction. So from the cytoplasm, a lot of granules will be given out. So this will secrete a lot of proteases. So it will harden the zona pellucida, and the sperm receptors in the zona pellucida will be destroyed. So the zona pellucida does not accept any more sperms. One enter, it shuts off the door. No more sperms allowed. Number 2, resumption of second meiotic division. We said it's still arrested in the second meiotic division. Then now it will resume the second meiotic division. It forms a definitive oocyte, and the other one is the second polar body. The definitive oocyte will have haploid number of chromosomes called as female pronucleus. Then we have the metabolic activation of the egg. So the sperm will carry some metabolic activation. So this metabolic activation will activate the female egg. So that is the result of fertilization. So the phases is entering the corona radiata, entering the zona pellucida, and entering into the fusion of the cell membranes. When the sperm enters, the oocyte will produce cortical zona reaction. It will resume a second meiotic division and it will get activated. So if you see in this picture down which I've given, that is the fusion of the male and female pronuclei. At this point, we cannot identify which is male and which is female. So eventually, we are all the same. So this is the life lesson I usually take. Eventually, when in a family, it is not that I am the bigger one or you are the bigger one. Eventually, when there is a fertilization, automatically there is no difference between a male and female pronuclei. We call it pronuclei because it is not still a haploid cell. Once they unite together, they form one nucleus, it becomes a diploid cell. So when it is a diploid cell, we call them as a nucleus. So the nucleus of the sperm also becomes swollen and becomes male pronucleus. So in this image A, you can see a female pronucleus is there, a sperm is inside. In the image B, both of them form pronuclei. In the image C, there is breakdown of the nuclear cell membranes and there is fusion. And in D, it's forming a zygote and it starts dividing to form a new baby. So this fertilization is where the male and female, they will form just as the nuclei or the pronuclei, and they will unite together, break down our cell membranes to form a zygote. Am I clear until now?
Yes, doctor.
Okay, yeah? So the fertilization means not just fusion of the male and female gametes. Fertilization means fusion of the male and female pronuclei. So if as a non-medical person, they will be telling, oh, fertilization is just fusion of male and female gametes. No, only medical people will know it's the fusion of male and female pronuclei. Okay, so we are one step higher always from the non-medical persons because they don't know this process. Like how we don't know what's happening in a machine, they what's happening in this. So when we read medicine deeply, we will know the intricacies. So that's how we expand our knowledge. So the results of fertilization. So what is the result of fertilization now? It restores the diploid number. So the male pronucleus has 23, female pronucleus has 23, fuse form 46. So now the zygote has 46 chromosomes, so it is back to normal. It has got 46 totally. It determines the gender of the fetus. So X carrying sperm produces XX embryo and Y carrying sperm produces an XY embryo because the female is always X. So the genetic determining factor is always the male gamete. It is never the female because female have only one X. So the person who decides, or the organelle that or the cell that decides the genetic the gender of the baby is usually the sperm. If a sperm containing Y fertilizes, it forms an XY. If a sperm containing X fertilizes, it's an XX. So automatically, once they fertilize, a zygote is formed, they immediately start dividing by mitosis. Because without fertilization, the ovum degenerates in 24 hours. So once a zygote is formed, it immediately starts dividing rapidly. Am I clear?
Yes, doctor.
Okay. So this is just a picture to show you. There is corona radiata. The zona pellucida is the brown color A, the figure A. The zona pellucida is brown color. There is one oocyte with the one polar body there. So when the sperm enters, by the time the ovum already forms a pronucleus. Going to the figure C, there is a female pronucleus and male pronucleus. Here they have shown that there are two colors, one purple, one brown, but in real life we cannot identify the difference between a male and female pronucleus. By D, there is a breakdown of nuclear cellular membranes. They have fused. Once they have fused, by E, they start dividing. So one cell becomes two cells. So this division occurs very fast within 24 hours after fertilization, because if the oocyte is not fertilized, it will degenerate. So once it's fertilized, immediately it starts dividing. So this division is called as cleavage. After the two-cell stage, the zygote will undergo mitosis. Cells increases, but the size decreases because they become more and more compact. They remove extra fluid away from it and becomes more compact. So these cells are called as blastomeres. I said blast means they are basically young cells. So they divide very rapidly. We call them as blastomeres. So after the 8-cell stage, so 1 forms 2, 2 forms 4, 4 forms 8. After the 8-cell stage, the blastomeres undergoes compaction. Compaction means you can see they form a closed ball of cells. 3 days after fertilization, they will form the 16-cell morula. So if the fertilization occurs on the 15th day, by 18th day, the baby is already 16-cell big. Here again, I don't go by the numbers. I don't want a 15 day, 16 day, no. I want you to remember the division starts immediately after fertilization, because if not fertilized and if it is not fertilized, automatically there is degeneration of oocytes. So once fertilized, they immediately start dividing and go to the next stage. So you will start forming the zygote. Then from the zygote, immediately the cleavage and compaction, forming blastomeres and eventually a morula. Who is Nurul Shahira Binti Mohammed? I think she's asking something, is it?
No, doctor. I mean, my mouse tertekan that button.
Oh,
Sorry.
okay. So then now we have a 2-cell stage. This is the 4-cell stage, and that will be the 16-cell stage. So it is still present within the zona pellucida. Zona pellucida is still holding the cells together. They just cannot move unless and until the implantation occur. The zona pellucida is still a firm protein coating that surrounds the zygote. So once the morula enters the uterine cavity, so now the fertilization has occurred in the fallopian tube. The 16 cells are already formed. They're slowly moving, and by around 5 days after fertilization, by the 6th day after fertilization, the morula will enter the uterine tube. When they enter the uterine tube, there is a lot of fluid secretion into the zona pellucida. They will form a cavity. So this cavity is called as blastocele. So all the cells are pushed to one end. So once they push to one end and you form a big cavity with fluid, if we see such an embryo, we call it as a blastocyst. It is at this stage the implantation occurs. So around 5 to 6 days after fertilization, the compacted cell will secrete fluid. The fluid will be pushed to one side and the cells will be pushed to one side. So it forms a blastocyst. By this time, the uterus is very ready. So in the uterine cycle, the uterus will undergo 3 phases. The proliferative, which we saw, which is the estrogen phase. Then ovulation occurs. So when ovulation occurs, automatically the uterus go into secretion. The green color represents the glands. The red color represents the blood vessels. So the glands are ready to pour their secretions to nourish the receiving zygote, and the blood vessels are fully loaded with oxygen, and they have a lot of nutrients to nourish the egg. So the zygote will implant in the endometrium during the secretory phase because it will receive all the nutrition from there. So at this stage, the uterine glands and arteries are coiled. The tissue is very succulent. You know what succulent? Like a celery. When you cut into a celery, fluid comes out from the celery, right? So that tissue is called as succulent. So meaning it's very soft and supple, and you have lot of secretions. The mother usually she retains, there's a lot of waterlogging, there is a lot of water retention, just to make sure that the uterine tissue is well supplied with all these nutrients so that it receives the zygote and nourishes immediately. So that's the reason progesterone phase we have a lot of water retention. So now the uterus is ready. So now implantation would occur. So when implantation occurs, so the first image when you see onto the left side, you can see the oocyte is released from the Graafian follicle. So you have primordial, primary, secondary Graafian follicle, oocyte release and formation of corpus luteum in the ovary. Okay, then it is the egg is taken up by the fallopian tube. Sperms come and they will actually fertilize. Once they fertilize, a zygote is formed, 2-cell stage, 4-cell stage, 16-cell stage, formation of a cavity and formation of a blastocele, and it gets attached to the endometrium. Look at the endometrium, the full of glands and blood vessels ready for the zygote to come. So now when the blastocyst wants to implant into the wall of the uterus, then only the zona pellucida will detach and disintegrate and allow the whole blastocyst to latch on to the endometrium. Clear? Is it clear?
What else?
Sorry, yes.
Where do this everything that's disintegrating? Where do they go to? Do they come out of the lady, or they still stay inside?
It's still inside.
You said
come out where?
like there's the zona pellucida detaches and disintegrates, and then we see the other things.
Yeah.
There are other things which also disintegrate. When they disintegrate, do they remain inside or they are?
They remain inside only. The glands will secrete a lot of mucus. So the lady usually has a white discharge throughout her menstrual cycle. It will come out through the discharge.
Okay.
Okay. Yeah.
Thank you.
So implantation. So if you would say implantation, the purple or the pink color, the whole zygote along with the cyst, they will attach or implant into the anterior or posterior wall of the uterus. So the whole zygote will enter into the endometrium. So we actually burrow the way the zygote will enter and the endometrium will close off. So it is not attaching to the wall. The whole baby is inside the wall of the uterus, or the endometrium is the innermost layer of the uterus. So that is where it is actually ready to receive all the zygote. So the zygote will actually move into like a parasite moving into the wall, and then the wall will close off. So basically it is not, we are just attaching, we are entering into the wall of the uterus and we are going and sitting will happily inside. So approximately 7 days after fertilization, in between the opening of 2 glands, the whole zygote will enter into the wall of the uterus to sit in there happily. So that is the process of implantation. So what will be the clinical correlates of this? So I have a video on this. Do you want to watch the video on fertilization? First, let me show you the video. Where is my video?
About 300 million sperm enter the vagina.
Can you see the screen, Noah?
No, doctor.
Good job. Can you see now?
Yes.
Can you see, but can you hear the voice? No.
No, without the sound.
That's good job, huh? Now, okay, wait, huh?
Fertilization is the epic story of a single sperm facing to unite with an egg and form a new human life.
It's about fertilization.
It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. Causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
Is it clear?
Yes, doctor.
Very clear. Okay, I have another video on implantation, just less than a minute. I can just show you that as well. Can you see this?
Yes,
Can you
doctor.
see? Okay. This video, I don't think so. It has a sound. It just shows you the implantation. So once the zygote is formed and it's pushed towards the uterine cavity, it divides in its way. It forms the 2-cell, 4-cell, 16-cell morula stage, and it wants to implant. So here, if you would see, this is the morula, which comes towards the uterine cavity. So now the morula becomes more and more increases in size. So it usually gets detached from the zona pellucida. So when it detaches from the zona pellucida, it will get attached to the wall of the uterus, it forms a cavity or a blastocyst. Don't forget about all these parts, just you know that there is a cavity. It enters into the endometrium and it stays within the endometrium of the maternal, of the mother. So actually it's not just attachment, it is entering of the whole zygote into the wall of the uterine cavity. That is what we call it as implantation. Clear?
Clear, doctor.
Okay.
Yes, doctor.
Okay, so I will continue with the clinical correlates. So we have already seen that there is a fertilization occurring. What are the phases?
Excuse me, madam.
Yeah,
Sorry.
yeah.
Sorry for the interruption.
Yeah, sure.
I've seen in a lot of lectures there are microtubules.
Yeah.
Are they the same as centrioles?
No, microtubules are the thin wire-like substances which tend to pull or push the substance, the organelles. Like for example, the nuclei. Centrioles are formed in the poles. Like in the North Pole, South Pole, there are two centromeres or centrioles which are formed on the side. These centrioles give rise to microtubules. These microtubules will pull them. So the centrioles are static in their position. Microtubules can move around and pull or push substances within a cell. That's the action.
Okay, thank you.
So clinical correlates. The ovulation pain can occur in females. I'm not talking about the menstrual pain. I'm talking about ovulation pain or pain during ovulation. It usually occurs during the mid-cycle when the lady is ovulating. This pain is called Mittelsmers. It's a mild crampy pain in the lower abdomen in the middle of a cycle. We know that LH is secreting collagenase or increasing the activity of collagenase to release the oocyte. There is muscular contractions of the ovary to push the oocyte outside. So this induces this mild pain. There is an increase in the basal body temperature of the lady as well. So at the time of ovulation, the hormonal changes, especially the LH surge, the estrogen high level, the raise of basal body temperature, automatically the cervical mucus or the white discharge which is being secreted in a female, usually the female genital tract or the uterus and the cervix, they have a lot of glands. They keep on secreting white color discharge, which is actually a mucus or a lubricant they secrete. During ovulation, this becomes very watery. It becomes very watery. The reason is the watery secretion will allow the sperms to swim and come towards the egg. Before ovulation or after ovulation has occurred 24 hours, when the oocyte dies away, the secretions again becomes very thick for a female. So at that time, it's very difficult the sperm to navigate and go in because it's already no use. The oocyte has already dead. So during ovulation, the hormonal changes will rise up the basal body temperature, and the cervical mucus or the mucus or which is being secreted from the uterus of the female is very watery, so it will allow easily the sperms to swim through to the oocyte. So when a couple is being treated for infertility, they usually take the basal body temperature method, or sometimes if they take the cervical mucus and they put it on a slider, you can see as a fern-like pattern of the mucus. It's a very old style of identifying. Now that there is ovulation kits are available which can tell the female has ovulated or not, depending on the hormonal changes. So in ovulation, women can experience a mild crampy-like pain because of the uterine or ovarian contractions. So this pain is called as Mittelsmers. It's Mittelsmers, L-M-I-T-T-L-E la. My spelling is wrong, sorry. Then coming to after fertilization. So after fertilization, I told you the embryo will not just latch on to the endometrium, it will burrow its way into the wall of the endometrium and go and sit inside. In this process, some blood vessels will be ruptured in the wall of the uterus. So imagine a lady, she has the oocyte has been fertilized on the 15th day. By the 21st or 22nd day, the zygote is actually swimming towards the uterus. When it swims towards the uterus, by 25th or 26th day, it starts burrowing inside. It will start moving inside the wall of the uterus. When it moves inside, it will rupture the blood vessels on the wall of the uterus and some bleeding might occur and there will be some abdominal cramps. But the lady might think it as menstruation has occurred. By the way, it is not menstruation, it is only implantation period bleeding. So how do you differentiate? It occurs only for a few days, very lighter bleeding and mild abdominal cramps. The ultrasound examination is usually normal when we do at this stage. So this is called as implantation bleeding. So these techniques of ovulation and fertilization and implantation has been helpful for us doctors to come out with assisted reproductive techniques. We call it as art of producing a baby or assisted reproductive techniques. Infertility can either be due to male or female factors. So ART has been developed to aid the infertile couples. So we have 4 different types, 2 which are in vivo, which takes place outside the body, and 2 are in vitro, which takes place inside. I'm sorry. 2 in vivo, 2 which takes place inside the body, and 2 in vitro, which takes place outside the body. So coming to in vivo first. In vivo is, number 1 is artificial insemination. Usually the sperms are retrieved, or in some countries where donation of sperms is legalized, the person takes the donated sperms and they are placed in the uterus at the time of ovulation. We measure whether the lady has ovulated through her basal body temperature, to her ovulation kit, or we look through ultrasound also. We can identify whether there is a corpus luteum there in the ovary, so we know that automatically she has ovulated. So in that case, they will release the sperms into the female genital tract directly. This usually is very useful when the cervical mucus or the secretions in the female genital tract is hostile to sperms. So when it's hostile, whatever sperms they release, it might not help, it might die in the way. So they will directly release into the very nearer to the fallopian tube so that they can fertilize very rapidly. So this is the first technique. The second technique is gamete intrafallopian transfer. Here, the sperms is retrieved or donated again, and oocyte is retrieved transvaginally. They retrieve the oocyte. Both of them, they will mix it and put it into the healthy fallopian tube via laparoscopy. Now you can ask me, why are even taking out the oocyte and putting it back again? So in this condition, usually when we fertilize it inside the body, the amount of defects in the baby is very less. So as much as the natural fertilization occurs in the fallopian tube, it is it's very good for the baby because it's occurring under natural conditions. So when it occurs outside the body, as in vitro, the chances of congenital anomalies, rejection of the pregnancy or termination of the pregnancy is very high. So we try to make it as physiological as possible, closer to the body condition. We have the in vitro technique. So if the mother cannot conceive just by in vivo techniques, we go to in vitro. The oocytes are recovered by laparoscopy, and the sperms are taken up from the donator or the retrieved from the male. They are placed in a culture medium. They form a zygote. Already it's right under our nose in a laboratory, and then they take the zygote and they are transferred into the fallopian tube within 24 hours. So we ensure that fertilization has occurred, and then we put it back into the fallopian tube. In case there is problem with a thick zona pellucida, the eggs are impenetrable eggs. They are very difficult to penetrate. The oocyte is pierced and the sperm is directly released into the egg. So that we do, even inject the sperm into the oocyte. This is called as ICSI, intracytoplasmic sperm injection. This is a very hi-fi technique, but still happens. So we decide the patient has what. So with the amount of eggs available, with the amount of sperms available, they decide whether they want a donated sperm or they take it from the same father. Then they decide they want to go for artificial insemination or for gamete intrafallopian transfer. If that does not happen, they either go for ZIFT, zygote intrafallopian transfer, and ICSI or intracytoplasmic sperm injection. These two techniques are done in vitro or outside the body. So, I mean, usually when we want to go for infertility treatment, we try whatever can be done in vivo first, and then only we go for in vitro. So these are the most common techniques they use for assisted reproductive techniques. Clear? So far clear?
Yes, doctor.
Okay.
Yes, doctor.
So the hello. Okay. I'm continuing with the implantation. So abnormal implantation apart from the anterior or posterior wall of the uterus. If it implants below or it goes to the cervix to implant, implants in the ovary, implant in the abdominal cavity, anywhere else apart or in the fallopian tube, we call it as ectopic pregnancy. Since most of the since the fertilization occurs in the ampulla, some chances are that the implantation occurs in the ampulla itself of the fallopian tube. So the fetus may grow up to 5 or 6 weeks only because the fallopian tube is a tube. How long can it stand the implantation? So it will rupture and it can cause very severe bleeding. This bleeding can extend into the surrounding arteries. Mind you, the uterus is very vascular because it is a pregnancy. It is very, very vascular. So any rupture can produce life-threatening bleeding and the patient may die in hours. 1 or 2 hours, the patient may collapse. Such massive bleeding can occur. So this is always when a lady is pregnant or when we do a PREGCA test. After that, we make sure that she goes for an ultrasound to check whether it is an intrauterine pregnancy. If there's not an intrauterine pregnancy, we do not find a gestational sac in the uterus, we find it elsewhere, it becomes an ectopic pregnancy. So we usually tend to terminate such pregnancies because it can rupture the area of implantation and cause life-threatening bleeding. For my class today, any doubts?
Doctor, how does this happen? Why does it happen?
Ectopic?
The abnormal implantation.
Okay, usually as I told you, the oocyte does not move with the help of flagella, they have to move by the cilia. Ectopic pregnancy is very common with advancing age, as well as when there is a lady has a history of pelvic inflammatory disease. When the fallopian tube has disease, it they usually produce loss of the cilia and replaced by fibrosis. So the cilia, whichever is there, it is actually the cells are fibrous and there is no cilia there. So basically there is difficulty in pushing the zygote through the uterine cavity. So it stays there in the tube itself and it implants there itself. That's the reason.
Okay, thank you.
Yeah. Any other doubts in this?
Madam.
Yeah.
I'm curious about the pregnancy testing kits. The one that, I don't know, you place a finger on it, then it tells you whether you are pregnant or not.
Is it? Do you have finger? The one I know is only urine pregnancy test.
There are another ones I've seen in movies and such places.
In movies, okay, maybe I will look through, I will let you know.
Okay.
Okay, I will
Or
let you know
maybe I
what's
saw my answer.
Yeah, maybe on Friday you just tell me in my class what is it, or I can look through also, no problem.
Okay.
Any other doubts? If no doubts, you can leave the class. I think you have a quiz at 11:30, right?
Yes, Dr. 11:30.
Okay, so I will come for the discussion at 12. I think Dr. Atika, once you finish the quiz, she will call me over for discussion. I will be in. So any doubts?
No, doctor, I think.
Okay, so thank you all. So please make sure you're there for the quiz. I think it's from the topics nervous system, nervous tissue, skin, and I have given one question from gametogenesis as well, which you know already. So just for the revision's sake. So see you all during discussion of the quiz, inshallah. Okay, I'm just leaving the class, huh?
Okay, thank you, Doctor.
Okay, thank you.
Thank you, doctor.
Thank you, doctor.
Thank you, doctor.